Hello everyone. We're here at Cold Spring Harbor Laboratory for the 86th Cold Spring Harbor Symposium on Quantitative Biology. The topic of this year's symposium is genome stability and integrity. My name is Peter Sherwood. I'm a molecular biologist, and I'm joined today by Shelia David, who's a professor at UC Davis, and she'll be presenting her work later this week during the symposium, but I'm delighted to have her with me here today to discuss what she's found. Welcome, Shelia.
Yeah, thank you, Peter. It's wonderful to be here.
Great. So maybe you can start just by giving us a little bit of background about your field and what your research is about.
Yeah, so I'm working on this really unusual enzyme called MUT-Y, which recognizes a damaged base, this 8-oxoguanine, that's mispaired with adenine, and this happens because OXO-G mimics thymine, and so these adenines become misincorporated, and then that leads to mutations. And I started out actually just, I'm a chemist by training, just wanting to understand how the enzyme works. I thought it would be important in cancer, but at the time we originally started, there wasn't a cancer connection, but so we were working on how the basic enzymology of how the enzyme functions, and then really exciting time was when there was we got a phone call from a geneticist in England who had found mutations in a family where they had this unusual polyposis phenotype in the colon that's it's a symptom that eventually leads to colorectal cancer, so it's a precancerous phenotype. And these patients normally have inherited mutations. If it runs in a family, they have mutations in this particular gene that leads to this polyps. But in this case, they had mutations in MUT-YH. And so that got us working on variants in MUT-YH and showing sort of the first connection between dysfunctional base excision repair and cancer. And now there's so many more connections that are coming up. So anyway, that's sort of where we started.
Right. And so the type of DNA damage that MUT-YH encounters, detects, and then is involved in repairing is caused by oxidative stress, which is a byproduct of normal physiology.
Right.
Because your body uses oxygen, and then you get radicals and hydrogen peroxide and other things that damage the DNA in this way.
Yeah, and also inflammation.
Okay.
So inflammatory responses, and that's perhaps why it's the colon may be particularly sensitive, is because inflammation in the colon may make the colon cells get more OXO-G formation than because this is in the case of MUT-Y, the disease or the MUT-YH variants, they're actually found in all cells, but the colon seems to be particularly sensitive, though it's also showing up in other cancers. So.
Okay, great.
Yeah.
So it converts this oxidative stress damages, it converts guanine into oxoguanine, which then forms, ultimately, it creates a GC to TA transition.
Yeah.
Okay, and that's the mutation that MUT-Y should repair if it's functional.
Right.
But in these families and in other situations where the MUT-Y protein is not working, then this damage persists and it leads to cancer and possibly other.
Yeah, I think the idea is that if you grow up your whole life with mutations not being repaired, then as time accumulates, the chance of hitting a really important gene, and and in this case, the APC is an oncogene. So once that gets inactivated, then the cancer goes crazy. Right. So yeah.
So now, in terms of your current work, you're looking at the various functions of MUT-Y, and you're dissecting them at atomic level, actually.
Yeah.
So can you tell us about what you're going to talk about later this week?
Oh, sure, yeah. So, we're really interested in these cancer variants, but we also realized that we didn't know that much about the protein. Even though we've been working on it for a long time, in some ways there's still a lot of things we didn't understand, and we had a hard time predicting, even when we looked at variants, whether they would be dysfunctional or not. And so one of the things we started thinking about is one key step that we don't understand is how the enzyme finds this OXO-G A base pair.
How it recognizes this faulty base pairing.
Yeah, because it's really just one atom. So the OXO, it's a OXO group adding to a particular position in the guanine. So it's really a small difference between guanine, and it's also very rare. It's 1 in 10 to the 6th.
Right.
And then when the OXO G, pairs with A, that's even less frequent, right, than even the OXO Gs, because sometimes it gets replicated correctly. So we started, using sort of our chemical background to make modifications of the substrate and really hone in on what piece of the OGA mismatch the enzyme is recognizing. And what we discovered that it really has to do with the unique structure that OXO G makes with A. And what it does is it positions one particular group on the guanine in the major groove side of DNA rather than the minor groove side. And that ends up being sort of the molecular signal that the enzyme recognizes. And so we imagine that the enzyme is like scooting up and down the DNA, but then it kind of bumps into this group and that stalls it.
Oh, okay.
And then it, rearranges, stays there and interrogates the base pair,
okay.
and then does the chemistry on the.
Oh, that's interesting to a non-specialist that this enzyme and maybe others are constantly just scanning and not finding things, and then they'll find something. It's not as if they're not bound and then they get there. It's that they're just scanning. That's good.
That they need to, otherwise.
And there's evidence for that, eh?
Yeah, that they scan the helix. Yeah, I mean, in mammalian cells, I guess, they may be brought to the DNA or to regions that, during DNA replication, but they still have to probably at least scan a local region, right, to find the lesion and really, like, clamp onto the damaged, mismatched base pair, right?
Right. So in terms of the part of MUT-Y that is involved in this recognition, earlier today I said MUT-Y is a busy protein, people. I counted just the following functional domains. There's RPA interaction, PCNA interaction, MASH6 interaction, APE1 interaction, HUS1 interaction, SIR2 and 6. There's an iron-sulfur cluster, and then the last one I wrote down was the one that you have been working on lately, which is the 8-oxoguanine recognition domain. So can you tell me about that?
Yeah, so one thing that's unique about MUT-Y is that it actually recognizes this damaged, mismatched base pair, but it's doing the chemistry on the normal base that's been sort of misincorporated, so it's sort of contextually wrong. Okay. So A isn't a wrong base if it's paired with C, but with OXO-G it's wrong. So the enzyme has to find these OGA pairs and then cleave out the A. So it has this special OG recognition domain that is important for recognizing the OG, but the two domains have to work in concert.
I see.
And I think once it's clamped on, that's when it, then is going to recruit some of these other proteins that you talked about. The AP endonuclease is the next enzyme in the pathway. That once MUT-Y cleaves the adenine base, then the AP endonuclease cleaves the sugar, the abasic site, and then a polymerase and a ligase come in. So it has, all these other factors have to be there to regulate its activity.
Right.
Because in some ways, what MUT-Y does, if it were just to cleave adenines, that without being under control, that would be bad.
Right. It has to finish the job by repairing. Yeah, I see.
Yeah.
So what do you think your plans for the future are? What are your next steps? I know there's many.
Yeah, so we're really excited about some of the tools we've developed, to answer some of these fundamental questions about now applying them more to studying cancer variants. Because one of the things we're finding is that each cancer, variant is different. And, genetics is wonderful, but sometimes people have a rare variant that that maybe it's only a couple of patients. Mm-hmm. And so that's where, having biochemical or cellular assays to give you some insight about what that variant might be doing is important. And then we're also learning about the protein in the process of studying these variants. But then another dream of mine is, at some point, is we're really interested in making small molecule inhibitors for MUT-Y and other glycosylases. And a lot of people think, oh, you wouldn't want to do this because why would you want to inhibit a DNA repair enzyme? But I think that they will be useful as tools, but potentially as therapeutics as well, because there's evidence that once you have cancer, then inhibiting DNA repair can kind of push you over the edge. So, in figuring out this sort of stop sign where the enzyme stops, that also led us to sort of discover this allosteric site in the protein that is far from the active site, that's part of this OG recognition domain that's specifically recognizing this 2 amino group that's sticking out. And so we think that's a good site to target a small molecule. So.
Oh, that sounds great.
Yes.
Yeah.
Yeah, so hopefully next conference I can talk about that.
Sure. Yeah. That's great. Well, thank you for joining me today, and I really enjoyed it.
Yeah, it was fun. Yep.